We are constantly told that a single exotic berry or a brightly colored pill holds the secret to cellular youth. The marketing aisles are flooded with products boasting about their ability to neutralize damage and reverse aging. We have been sold a compelling narrative that isolates specific compounds and elevates them to miracle status. But when we look closely at the biological reality, the truth about antioxidants and their health benefits is far more complex and fascinating than any label would have you believe. To understand this narrative, we first have to look at how our bodies generate energy. This brings us to metabolism, which refers to the complex chemical processes that occur within a living organism in order to maintain life and convert food into usable energy. As our cells perform these essential functions, they naturally produce byproducts. Among these byproducts are highly reactive molecules that play a central role in the aging process and disease development. Scientists call these unstable byproducts free radicals. A free radical is simply a molecule that is missing an electron in its outer shell. Because electrons prefer to exist in pairs, these molecules are chemically frantic. They scavenge the body, aggressively seeking out other molecules to steal an electron from, which creates a destructive chain reaction. When a free radical steals an electron from a cell membrane or a strand of DNA, it damages that structure and creates a brand new free radical in the process. Our bodies are well equipped to handle a baseline level of this chemical theft. However, environmental factors like pollution, ultraviolet radiation, and poor dietary patterns can cause these unstable molecules to multiply rapidly. When the concentration of free radicals overwhelms our natural biological defenses, we enter a state known as oxidative stress. This prolonged state of cellular damage is heavily linked by research to chronic inflammation, cardiovascular disease, and the broader mechanisms of biological aging. This is where the heroes of our biological story step in. Antioxidants are unique molecules that possess a remarkable chemical ability. They can safely donate an electron to a free radical without becoming unstable themselves. By offering up this spare electron, an antioxidant effectively neutralizes the free radical, stopping the destructive chain reaction in its tracks and protecting our cellular architecture from further harm. While our bodies manufacture some of these protective molecules internally, we rely heavily on our diet to provide the rest. We obtain these primarily through micronutrients, which are essential dietary elements like vitamins and minerals required by organisms in small quantities throughout life to orchestrate a range of physiological functions. Familiar compounds like vitamin C, vitamin E, and various plant pigments all serve as powerful electron donors within our biological system. The food industry quickly recognized the commercial potential of this biological mechanism. This realization birthed the modern superfood myth, a marketing strategy that elevates ordinary nutrient-dense plants to mystical status based solely on their laboratory-measured electron-donating capacity. We began seeing exotic fruits and expensive powders marketed as ultimate shields against disease, reducing the profound complexity of human nutrition to a simple, marketable metric. The logical, yet flawed, next step was to extract these molecules and pack them into pills. The assumption was that if a diet rich in these compounds is good, then massive doses of isolated antioxidant supplements must be even better. For decades, millions of people have consumed high-dose vitamins, fully expecting to bulletproof their immune systems and extend their lifespans based on this reductionist theory. Yet, as clinical data accumulated, a surprising paradox emerged that forced scientists to rethink everything. Large-scale, peer-reviewed studies examining the long-term effects of isolated antioxidant supplements repeatedly failed to show the expected health benefits. In fact, researchers were baffled to discover that in several major trials, individuals taking high doses of synthetic beta-carotene or vitamin E actually experienced higher rates of certain diseases and mortality. Why would a molecule that protects cells in a laboratory setting fail so spectacularly when isolated in a pill? The answer lies in a concept called bioavailability, which is the proportion of a substance that successfully enters the circulation when introduced into the body and is therefore able to have an active physiological effect. In nature, nutrients never exist in a vacuum. They are packaged within a complex matrix of fibers, enzymes, and cofactors that dictate exactly how our bodies absorb and utilize them. This brings us to the crucial role of the gut microbiome, the complex community of trillions of microorganisms living in our digestive tracts. When we consume whole plants, these resident bacteria actively ferment and metabolize complex plant compounds, transforming them into thousands of secondary metabolites. It is this intricate, symbiotic processing that unlocks the true protective potential of the foods we eat, a process entirely bypassed when we swallow an isolated synthetic compound. Furthermore, clarifying the science reveals that free radicals are not entirely villains. In moderate amounts, our bodies actually need them to function properly. Free radicals act as vital signaling molecules that trigger essential biological responses. For example, the temporary spike in oxidative stress during exercise is precisely what signals our muscle cells to adapt, grow stronger, and improve their internal antioxidant defense systems. When we flood our systems with massive doses of synthetic antioxidants, we risk blunting these necessary biological signals. By artificially suppressing all oxidative stress, we might actually prevent our bodies from making beneficial adaptations. This explains why athletes who consume high doses of vitamin C and vitamin E after training often see a reduction in their cardiovascular and muscular gains compared to those who simply eat a balanced diet. Let us look at specific examples from the produce aisle to illustrate this synergy. Carotenoids are the vibrant pigments that give carrots their orange hue and tomatoes their deep red color. To properly absorb these fat-soluble compounds, our digestive system requires the presence of dietary lipids. Eating a raw carrot provides some benefits, but roasting that same carrot in olive oil significantly increases the amount of protective compounds your body can actually absorb and utilize. Similarly, the vitamin C found in a whole orange comes packaged with flavonoids and dietary fiber. This natural matrix ensures a slow, steady release into the bloodstream, maximizing cellular uptake. When you extract that vitamin C and compress it into a massive 1,000 milligram pill, your body simply cannot process the sudden influx. The vast majority of that isolated supplement is rapidly excreted, providing little more than expensive waste. The most robust dietary patterns studied by scientists, such as the Mediterranean diet, do not rely on isolated megadoses. Their profound protective effects stem from the consistent daily intake of thousands of different phytochemicals working in harmony. The true power of these dietary patterns lies in the synergistic interaction between whole grains, legumes, vegetables, and healthy fats, creating an internal environment where our cells can thrive. Understanding this evidence empowers us to make smarter, more effective choices about what we consume. We can confidently step away from the marketing hype of the isolated pill and overpriced superfoods. By embracing a diverse array of colorful whole foods, we provide our bodies with the exact complex chemistry they evolved to utilize, proving that the most advanced health interventions are often found right in our kitchens.